All right. Okay. So we did some brief introduction for classification of protozoa. For each species, for each genera, I would say that we're going to be talking about, you basically need to know all that same stuff. Something like route of transmission. I can announce up front there will be no vaccines, so don't worry about this thing. Route of transmission, generally where you're going to find it, what kind of disease, and what class does this microorganism belong to. Like Mastigophora, Amoeba, ciliates, apicomplex, and stuff like that. We're good? Okay. So first, we're going to chat about some amoebic microorganisms. The first ones are Acanthamoeba and Balamuthia. So these two genera of amoeba normally inhabit soil. However, they can enter the circulation through the broken, scratched skin or via the conjunctiva of an eye. Upon the entry, they get into the blood and eventually migrate into the brain, causing meningoencephalitis, meaning that's inflammation of the meninges, inflammation of the brain parenchyma. Now, you may remember that another species of amoeba that can cause meningoencephalitis, Malaria fowleri, caused the condition called primary amoebic meningoencephalitis, or PAM. Balamuthia and Acanthamoeba cause granulomatous amoebic meningoencephalitis. With a multiple, here you can see multiple granulomas located to basal nuclei. Now, what's the difference between primary and granulomatous amoebic meningoencephalitis? Mainly in the way the infection goes time-wise. Primary amoebic meningoencephalitis is extremely rapid, the one that we talked about in the very beginning of the semester. This one, the granulomatous one, is more slower. So there are clinical reports. Actually, I listened to a presentation of such clinical report when a doctor, American doctor, who was volunteering down in South America. I can't tell you exactly which country, I don't remember, but he was a volunteer there, and mother brought her two kids, son and a daughter, to this physician, saying that, you know, they kind of act weird, especially son has headaches and his behavior altered significantly and his sleep pattern changed, and he gave diagnosis very quickly because one of the signature, I don't know, signs, symptoms, kids usually acquire this infection by, you know, crawling around on their knees, you know, when they play on the ground, on the soil. Their knees are scratched, amoeba gets into the circulation, and it causes a very specific discoloration of the skin at the site of entry. So if you notice the discoloration pattern, told mom they've got this infection. It was lasting for like a couple of months. So kids got treated with amphotericin B, recovered. So if it's treated timely, the patients with granulomatous amoebic meningoencephalitis can actually recover. Okay. However, as you can imagine, this infection is more common to tropical developing countries, so getting proper medical care timely sometimes is problematic. Another amoebic microorganism, Entamoeba histolytica, which life cycle is shown here, is a main cause of gastrointestinal amoebiasis. The transmission is via food, drink, or any other kind of contaminated water, okay? And this microorganism gets into the large intestine, bypassing the small one, and releases the enzymes. Exoenzymes destroy tissues, leading to ulcerative colitis. So these ulcers, ulcers in the digestive tract may lead, well, first of all, that's going to be diarrhea. Does that make sense? Because damage to the mucosal layer of the colon will inevitably lead to the poor absorption and problems, with the normal ion absorption in the colon. Second, since those are ulcers, that may lead to the bleeding into the colon and leading to the dysentery. In some cases, it even leads to perforation of the intestine and may cause peritonitis. Now, this is gastrointestinal amoebiasis. You can see that all these symptoms here are localized to the colon. Now, there are examples of extraintestinal amoebiasis. Microorganism, upon the entry into the circulation, can get into the liver and the lungs. And it's actually quite interesting because we're going to have another microorganism that can cause liver formation of the liver masses, Echinococcus granulosus. So far, what I want you to appreciate when amoeba enters into the liver, infects liver cells, it forms up a pretty large cyst. Does that make sense? Fluid-filled cavity, okay, which can be actually tapped, and the fluid can be removed, and amoeba can be identified in that fluid, okay, and it also can enter by circulation, can infect lungs, leading to the formation of so-called amoebomas, okay. So amoeboma looks like a granulomatous lesion, okay, and on X-ray, the pulmonary amoebiasis looks like lung cancer. And there were examples of patients coming in for evaluation because they have respiratory distress due to the amoebic infection of the lungs. They would get chest X-ray, they would be diagnosed, quote unquote, with lung cancer, and then physician would do the biopsy of the suspected tumor. They will come back and say, well, congratulations, you don't have lung cancer, you have pulmonary amoebiasis. It's always better because it's treatable. Now, how important amoebiasis is worldwide? Very much. Well, amoebiasis only contributes to about 100,000 deaths worldwide. Now, this infection affects as many other protozoan infections, mainly developing countries in the tropics, where simple lack of proper running water and the proper sewage contributes to the contamination of the drinking water with amoeba. Because the life cycle here, life cycle here, as you can see, is humans plus cysts and trophozoites. Cysts and trophozoites can ingest it back. So it's basically human to human. Does that make sense? Now, there's no way we can cover all interesting protozoa due to the limited time. But I want to mention that if you are going camping and you know that you're not going to be able to bring probably all the drinking water with you, especially if it's like a hiking trip. There are three simple ways to make sure that water that you collect from the stream or from the lake is potable. One is to boil it. That's the simplest one. Boiling will destroy all protozoa, cysts, and trophozoites alike. And it will destroy the overwhelming majority of bacteria that can cause gastrointestinal disease. And it doesn't alter the taste of the food, the taste of the water. Second is iodine tablets or some other cleaning tablets that may change the taste a little bit. And filtration, you can buy actually filters. They work pretty well. There are filters that you just hang on the tree and it, just trickles down, or you can forcefully filter it through using pump. That works. Okay. In my opinion, like if there's firewood and there's plenty of it, then you can do the bonfire, just boil it. Now that is unarguably the most medically important protozoa that we know of, malaria. Well, malaria is the disease. Protozoan is Plasmodium falciparum. It's apicomplex, meaning that it's obligate parasite. Okay, and there are multiple species: Plasmodium falciparum, Plasmodium vivax, ovale, Malariae. You don't need to memorize the names of the species. You do have to recognize the name of the genus. So, for all of these species, the biological vector is a mosquito from the genus Anopheles. Sometimes they're referred to as Anopheline mosquitoes. For instance, in Africa, that's going to be mosquito Anopheles gambiae. Now, what's so different between these four or even more species of Plasmodium? Geographical distribution. For instance, Plasmodium malariae is found mainly in Southeast Asia. Plasmodium falciparum, Plasmodium vivax are pretty abundant all over the world, from South America to Southeast Asia. Does that make sense? So that actually, geographical distribution is the first. Now, how important medically it is. To be honest, you look at the data, even in the peer-reviewed papers, they will be all over the place. Not too much, they're not going to vary from 100,000 to 100 million, but almost half of the world population live in the malaria-affected areas. Okay, every year about 200 million cases and 500,000 deaths. I believe I collected this data from WHO, but it's 2013. It's better now. We're getting better every year. By a little, but better. So first, what happens? What happens during the actual clinical disease? You get bitten by a mosquito. After about 2 weeks, you develop the primary symptoms of the infection, being fatigued. We have someone who can tell how it feels. Do you want to tell, Jacob?
I mean, it's just gross. There's nothing else to say other than it's just really gross.
Awful.
You feel awful, and diarrhea is just...
So yeah, the presentation that is described, presentations may vary from person to person. The presentation that's described here in this slide is kind of general symptoms that virtually everybody would feel, like aches. Very common is the backache in case of malaria infection. Now, after that primary story, you can experience the recurrent bouts of chills, fever, sweating, and shaking every so many days. Now, here I provide the interval of 2 to 3 days. In fact, depending on the type, meaning whether it's Plasmodium vivax or Plasmodium falciparum or Plasmodium ovale, the interval of the symptoms will vary. I can never remember, which one's which, but let's say for one of them it's going to be every 3rd day, for another, and it's very, very like clockwork. Every 3rd day for one, every 4th day for another, every 2 weeks for another. And basically, based on the symptoms, you can fairly accurately predict what species infects a particular patient. Does that make sense? Now, now the problem is that this microorganism can actually, it infects red blood cells, and if it is left untreated, microorganism will lead to the vasculitis and obstruction of the blood vessels in the brain, leading to the impaired blood flow to the brain, oxygen deprivation, and eventually coma. This is called cerebral malaria, when malaria basically stays in the cerebral blood vessels. Cerebral malaria is really hard to treat, and it's usually lethal because it's kind of too advanced. Does that make sense? Now, the population that is mostly at risk in Africa are children, age from about 6 months to 5 years. Up until age of 6 months, maternal antibodies protect them from malaria pretty well. At least not from the infection, but from getting sick. And basically the idea is, if child survives the age of 5, they're going to be fine. Okay, they're going to grow up. Are they going to get new cases of malaria? Absolutely, yes. They will be reinfected over and over again. But with every new infection, the immune system will receive another boost. So in adults, often the malaria infections become milder and milder with age. Some kind of sort of a natural immunity forms, you know, but surviving until the age, over the age of 5 is kind of problematic. Does that make sense? So it's treatable. A whole bunch of drugs. Here I list only quinine based. So quinine was an anti-malarial drug isolated from the bark of some South American tree, which local folks identified as the drug way before any pharmacology started in Europe or anywhere else. So quinine was isolated for a long time. It was considered to be one of the most bitter compounds on earth, super bitter. Then it was chemically modified, so chloroquine, quinidine, mefloquine are semi-synthetic derivatives of the quinine. In 2015, Chinese female scientists received a Nobel Prize for identifying a novel drug against malaria called artemisinin. It's pretty fascinating story. She did she's done an amazing approach. She knew that in ancient China there was a treatment for malaria, and that it was derived from plants. So they went back to read the ancient Chinese manuscripts on how to treat malaria. She identified the plant, she isolated the compound, and that was artemisinin. I mean, we already have a bunch of plasmodia that are resistant to different quinines and, artemisinin, but nevertheless, I think it's pretty exciting. She was the first female Chinese scientist and first female Chinese scientist to receive Nobel Prize. And she didn't have doctorate. She had bachelor's only. So that was a pretty cool story. Now, so treatment available. Any vaccines? Interestingly enough, no, there are no vaccines. By the way, side effects. You were treated for malaria? What are the side effects? Malaria treatment. Mefloquine.
I had visual and audio hallucinations, nightmares.
Yes.
Really messes you up.
Yes.
Don't do it.
Yes, that's so one of the side effects officially listed is vivid dreams. It's kind of a nice way of saying what Eric mentioned. Yeah, hallucinations, and there are some people, I read the stories of patients who said, we came to Africa and you have to take it prophylactically. They started taking it and they said, screw that. I'd rather have malaria.
Yeah. Okay, you say that.
Hmm?
You say that until you get malaria, and they're like, okay, the side effects are not that bad.
True, yeah. But the idea is, it's not just, you know, popping a pill once a day or whatever. It's really serious side effects that really makes you feel bad. Now, treatment, vaccine. Interestingly enough, you can study malaria vaccine on humans. Officially. There are studies like that. You can make a vaccine, you can vaccinate a person, and then, what's important, you can give that person malaria. You can infect them with Plasmodium. It's allowed. Because it's completely treatable infection. There were studies of the vaccine done on humans, and there is a vaccine candidate for, you know, about 30% effective. With the infection like that, medical professionals will take 30% efficacy. Basically, the idea is that you're going to have 30% less kids who will die. And absolutely, if we're talking about, you know, instead of 300,000 children, we have 200,000 children dying, sure. Okay, everybody will take it. Now, if you would look at the pattern of infections, the mosquito-borne infections in the world, you may notice that up until early 70s, I would say, the mosquito-borne infections were pretty rare. There was no huge outbreak of malaria in Africa. There was not a lot of dengue virus infections or West Nile or yellow fever. And then suddenly in the 70s, they skyrocketed. Any idea what happened in the 70s? They banned something. Yeah, they banned DDT, which was sprayed from the planes to control mosquito population. So when pesticides like this were banned, mosquito population boomed, and, you know, we've got a lot of transmission. So now the main way to reduce the number of malaria infections is to prevent the transmission. Basically, Melinda and Bill Gates Foundation, they spend a whole bunch of money in Africa on this, bed nets and repellents. They equip cribs for children with bed nets, and that immediately reduces the chances to acquire, because most of the bites happen during the night. There are some ideas on using bacteria called Wolbachia to control the population of mosquitoes. And don't get me wrong, but there is an idea to use DDT, not in the approach spraying it from the planes, but to apply it to, surprisingly, a windowsill and the frame of the windows in the houses. Because people sleep with open windows, they can't afford AC, right? And turns out the mosquito behavior is that it doesn't fly directly into the window and bites you. It actually lands on the window frame, windowsill, before it gets into the house. So if you could spray DDT on the windowsill, mosquito will land and will die pretty quickly. It was shown experimentally to work. But it's kind of, again, it's debated, because DDT got a pretty bad rap. Now, I want to chat a little bit about the life cycle. And before we, hold on, it's super intimidating picture. And I will tell you what I want you to know, very specifically. So first, I will not ask you about the names of the different forms, like merozoite, or schizoid, okay, or sporozoite. Here's what I want you to know. I want you to know that Plasmodium, after the mosquito bite, enters the circulation. You got it? The blood. Okay? By the way, the term blood meal, why is it incorrect when we apply it to mosquitoes? What do they eat? Mosquitoes. Huh? They're not eating blood. They eat sap. You can grow mosquitoes on, like, fruit juice. What do they need blood for? Babies, yeah. They need protein source for reproduction. This is why the only mosquitoes that bite you are female mosquitoes. Male mosquitoes don't bite. So, enters the blood. And from the blood, it gets into the liver. Okay? This is why one of the characteristic symptoms of human malaria infection is hepatomegaly, enlarged liver. Does that make sense? Now, Plasmodium replicates in the liver and then infects red blood cells. You can see the infected red blood cells. Hold on a second. So, infected red blood cells, you can see them right here, okay? So ring form and schizont, you don't need to know that stuff. And the point is, every so often, the Plasmodium cells, they leave red blood cells, you see, and lead to hemolysis. Every time that happens, every time they complete the replication cycle and leave the red blood cell, the patients have chills, fever, and sweating. Does it make sense? Now, here's the clinical connection for you. Each species of Plasmodium has different length of replication cycle. Is that understood? Do you understand what I'm saying? So, say one species will require 3 days to complete the blood study of replication. So every 3 days you're going to have this chills and fever. Another species requires 2 weeks. Every 2 weeks you have these bouts. We good? Now, so far, Plasmodium enters the blood, then infects liver, then red blood cells. In fact, another symptom of ongoing malaria is splenomegaly, enlarged spleen, not only liver but also spleen. The reason for it is that damaged red blood cells will be utilized. And which organ is responsible for the recycling? It's the spleen. When you have a whole bunch of damaged red blood cells, spleen works like in the 3 shifts, and it simply gets bigger because of that constant recycling effort. Good? Now, what else? One important stage that happens in humans is that some of the cells, of some Plasmodium cells, they actually go through the meiosis in the right bottom corner, and they produce gametes. Can you see the gametes? Or gametocytes. So gametocytes are acquired by the mosquito. When 2 gametes fuse, what is formed? A zygote, right? And zygote develops in the adult Plasmodium. Does that make sense? So you have a diploid Plasmodium that goes through all these stages. Okay? At some point, some of the diploid Plasmodium, they undergo meiotic division, produce haploid gametocytes, which then mate in the mosquito. We clear? Yes. Mosquito bites second time. It takes a blood meal and collects. So that's basically mixing. One of the approaches to reduce the malaria, there are genetically modified mosquitoes that bite only once. And that'll work, you know. They take 1 blood meal. So basically, who already infected, they screwed. But they can't transmit because there is no second blood meal. You see what I'm saying? So yeah, it's just multiple bites. When it bites, it collects the blood and collects gametocytes. Good? Now, what I wanted to say. Oh, look at this. So which organism is the... Where do the sexual cells develop? In a human, right? That makes sense. in which organism do they mate? In the mosquito. Correct? Because look at this. Look at the left. So, gametocytes mate and produce zygote in the mosquito stage. Does that make sense? So I'm going to introduce a term for you that I want you to know. Actually, two terms. In protozoan and helminthic infections, there are two types of hosts. I'm sorry for my voice. One is intermediate host. And another one is definitive host. So definitive host is the one in which the sexual stage of reproduction happens. Meaning, definitive host is the one where either gametocytes or gametes fuse, forming zygote. Does that make sense? For other organisms, which we'll get to, it's the host in which eggs are produced. So interestingly enough, for malaria, the definitive host is mosquito, and the intermediate host is human. Does that make sense? So this is like, humans are very anthropocentric. We tend to think that it's all about infecting us, killing us. But in this case, technically speaking, malaria is the infection of mosquito. Mosquitoes are definitive host for malaria. And humans are simply intermediate host, sort of, a link in that chain. The fact that we're getting sick, well, too bad. Mosquitoes, by the way, don't feel well too. Malaria does some damage to them. That's the idea that actually is discussed, to eliminate mosquitoes. The problem is they are extremely important food supply for a lot of amphibians. And disrupting the food chain will have a very long-lasting, far-going, unpredictable consequences for the entire ecosystem. So if we could make mosquitoes that bite only once, that would help tremendously. Okay, that make sense? Any questions on this one? Okay, another pretty cool protozoa, Toxoplasma gondii. It's another epicomplexan, meaning that it's obligate parasite. And this transmission, so that scheme on the right, I think it's a little bit overloaded. This is, by the way, Toxoplasma in the blood. Okay? So I'm going to talk about the transmission cycle and then some sort of social consequences of it. So how can you acquire Toxoplasma? By the way, it's the perfect parasite. Some estimates put that 1/3 of the world population are infected with Toxoplasma. As you can imagine, that's pretty badass, One transmission route is from cats. So, infected rodents, which will consume oocysts from feces, infected rodents will have a very interesting change in behavior when they acquire Toxoplasma and develop toxoplasmosis. They stop fearing cats. Mice, specifically, know where the cat is because they can smell the cat's urine. Turns out that when they're infected with Toxoplasma, they stop sensing cat's urine. They're not like becoming super cool and super brave. They just don't know that the cat is around. So that essentially increases the chances for a cat to catch a mouse, okay? And that catching a mouse and consuming the mouse will give an infection to a cat, which will pass cysts in the feces. That make sense? People, sorry, cleaning the cat litter box will expose themselves to the feces. There's a likelihood of contaminating the fingers or something like that, okay? And basically you need one cyst to establish the infection, so that those cysts will be passed orally to humans, and humans will develop the infection. Does that make sense? Clear? So from rodents to cats, from cats to humans. That's kind of, you know, in-house route of transmission. Second route is, well, contaminated foods, but let's admit we're not walking around spraying toxoplasma all over the place. So these contaminated foods probably, you know, like cats are around the house. Another route of transmission is from cattle or pigs. By consuming undercooked pork or undercooked beef, there is a pretty good chance of transmitting toxoplasma, you know, acquiring cysts, okay, and there we go, you're infected. So about almost a quarter of the US population are infected with toxoplasma. Now, in a couple of countries, like France and Brazil, Brazil is not here on the list, but the infection rate, the number of people, the percentage of people who are toxoplasma positive can be as high as 80%. And I can tell you that this is tightly linked to some of the signature dishes and national cuisine of France and Brazil. You know what Brazilian steakhouse is? They serve a lot of meat. Brazilians eat a lot of meat. And often, you know, this meat is served not raw, but medium rare, for instance. It doesn't kill all the cysts. So people constantly get exposed just by consuming significant amounts of undercooked beef or undercooked pork. And about 80% of population are infected. In France, the sort of national staples dish is steak tartare, which is practically raw beef, okay? And that's the route for transmission. Now, what it does to a healthy, immunocompetent adult? The answer is nothing. Really nothing. I mean, it can go to the brain and get encysted in the brain. That's it. Nothing's going to happen. It poses a significant risk for newborns, for a fetus, because toxoplasma can cross placenta. Okay, so pregnant women are advised to stay away from the cat litter and from eating grossly undercooked meat. Okay, and it is one of the main problems in patients with AIDS. Not HIV, just AIDS. It's a huge problem for AIDS patients. Now, the question is, since it affects the behavior of the rodent hosts, can it affect the behavior of humans? There were studies linking toxoplasmosis in immunocompetent individuals to behavioral changes like aggression and even, you know, road rage and schizophrenia. Now, those studies, if you review them, they're not rigorously controlled and very well performed, okay? And for me, the main argument against those, look, if 80% of France is infected, I mean, they're weird, but they're not schizophrenic or, you know, have a road rage or something like that. And the majority of scientists that actually study, you know, the molecular mechanism and epidemiology of toxoplasma, they I don't want to say believe, it's not about belief, but they say that those studies, they're not done well enough to suggest that there is an actual link. Okay. Basically, what does it mean for you if you have cats or if you really like a medium rare steak? Unless you get immunocompromised, you're totally fine. You can live long and prosper and never know about toxoplasma infection. Now, Leishmania is a pretty interesting microorganism. There are several species. You can see the world geographic distribution. The darker areas, the brown and dark red, are the areas with the largest percentage of Leishmania transmission. This areas basically where single cases may be reported due to some weird occurrence. Leishmania is transmitted by a scent fly. It's a biting insect, a relative of mosquitoes. It belongs to the family Diptera. When scent fly bites a person, it transmits Leishmania either in the skin, or the microorganism can enter mucous membranes or become systemic. So it's a mastigophora. Here you can see flagellum. Okay. Now there are three forms of Leishmania infection, or Leishmania disease. First is cutaneous Leishmaniasis or Chiclera ulcer. As far as I remember, now I wouldn't take these words to the bank. Chicleros was an argot word for field workers that worked very early in the morning. They went to collect something, do something in the fields, okay? So that's the time when scent fly bites the most. So they were working in the field, were exposing themselves to those bites during the highest activity of this insects, so they've got those ulcers. Ulcer itself is painless. It's not itchy. I don't know if you can see it. Let me try and do this. So that's the ulcer, okay? Interestingly enough, you can diagnose Leishmania by doing a scraping of the edge, because this is where the microorganism is. And in many cases, in some cases, it can actually, people can recover. If they're healthy, you know, like if you travel to South America, get Leishmaniasis, come back, you can recover. This ulcer can last for a while, okay? But interestingly, it's absolutely treatable, right? There are the drugs to get rid of. Make sense? Now, much worse form is mucocutaneous form, or spondia. Here, spondia is shown to be on the tongue. One of the main sites for mucocutaneous Leishmaniasis is the mouth and nasal cavity. And in untreated cases, mucocutaneous Leishmaniasis can destroy the structures. For instance, it can completely destroy soft palate, leading to, you know, pretty gross anatomical problems, and, you know, people cannot properly eat and stuff like that. And that has to be surgically restored, okay? Now, the most severe form is Kala-Azar. That's also called visceral Leishmaniasis. Liver and spleen are affected. Hepatomegaly, splenomegaly. If Kala-Azar is left untreated, that leads to death. Untreated visceral Leishmaniasis is 100% lethal. Now, one interesting story. If you ever had Leishmaniasis, you are deferred from donating blood. We talked about that in application for trypanosomiasis, both Chagas disease and African trypanosomiasis. Same is true for malaria. If you ever had malaria, you deferred from blood donation. And same is true for Leishmaniasis, because treatment is not considered to be sterilizing. Now, a pretty big problem arose in 1991 when American troops participated in the Operation Desert Storm in Iraq, freeing Kuwait. A whole bunch of American troops got Leishmaniasis, and it removed a pretty significant pool of potential blood donors for the army, because they were deferred for life. Now, here's what I suggest we do. We have about 40 minutes.